All right. So we are getting into this organic macromolecule business, and there's four that we're focusing on: carbohydrates, lipids, proteins, and nucleic acids. I'm going to buzz through some specific examples because I think that is helpful to get some better understanding of what these things are and how they function. So this is what the standard is to understand structure and function. So what are these things made of and what do they do? So basically you're going to be identifying them by their chemical structure and what they look like, and then explaining what they do for living things. So we're going to start with carbohydrates. Carbohydrates, again, as far as identifying, hexagons, pentagons, only carbon, hydrogen, and oxygen, no other molecules. And one example is amylose or starch. So we think of starchy foods. These are plants that store carbohydrates as they do photosynthesis and capture energy from the sun. They create some ATP and then they store energy in the bonds of sugars or carbohydrates. So carbohydrates can be monosaccharide simple sugars, one individual molecule like this, or they can be complex, like amylose. Again, if something ends in ose, we're thinking carbohydrate. So this whole thing is what we can eat and digest in order to get carbohydrates into our system. So when we finally break all this stuff down, we break it down into simple sugars, and glucose is an example of that. We split glucose, we split this sugar to get energy. Whenever you see this little ATP thing, we're thinking energy. And then we can take that split sugar and break it down a whole bunch more, use this membrane inside mitochondria to develop a lot of ATP. So carbohydrates are an energy source because we can take the energy stored in the bonds of the sugar and put them into bonds of ATP, which we'll talk about when we do cellular respiration. Okay, glycogen. When we have excess carbohydrate in our system, we also can store excess carbohydrates for energy usage later. And we don't store it as starch or amylose, we store it as glycogen. So we reattach all of these simple sugars back together to make a complex carbohydrate. And if you ever hear of like carbo loading, we are trying to maximize the amount of glycogen stored in our liver and in our skeletal muscles. And our liver is where we store most of our glycogen. Okay, let's move on to lipids. So, lipids, again, we often think of them as fats, more common name, but they do more than what we will talk about what fat is, but huge important lipid is a phospholipid. That's its structure. And its function is to serve as a membrane. So we have phospholipids because these fatty acid tails are nonpolar and repel water. So they are attracted towards each other, whereas the heads here are polar and are attracted to the polarity of the water. So the water inside and outside of cells are attracted to the head part of these phospholipids, and that creates a barrier between the inside of something and the outside of something. And we often think about the cell membrane or the plasma membrane separating the outside of every single cell, but there are also membranes, lipid bilayers inside around different organelles within our cells. So phospholipids, still glycerols and fatty acids, they do have a phosphate group on top, which is where the phospho part comes from. But lipids are typically carbons, hydrogens, and oxygens as well, except we have these long branches of carbon-hydrogen chains. Okay, this is where we actually think about fat, triglycerides and adipose tissue. We all have fat underneath our skin. So subcutaneous fat, we have our outer skin, our epidermis, our dermis, and then we have this area below the dermis where we can store fat. And we have these cells, and they have nuclei, but we pack in all of this extra triglycerides to store energy. And it's stored energy because we can take these fatty acids, we can take the free fatty acids from triglycerides, and we can put them into mitochondria and continue to break them down here and get a lot of energy. We'll talk a lot about that when we actually do cellular respiration. But these triglycerides in adipose tissue, so a function of a lipid is to actually store energy. Another lipid would be a steroid hormone, and hormones are communicators. So steroid hormones travel from endocrine glands through the bloodstream to target cells. One example of a steroid hormone communicator is testosterone. And again, it's carbon, hydrogen, and oxygen. Here we have a steroid hormone, and the idea of a steroid hormone is that when it binds to a receptor here, it actually goes into the cell. Since it's a lipid, it can get through this lipid bilayer easily, bind to the receptor, go into the nucleus where there is DNA, and this is where we turn on your DNA. This hormone receptor complex has the ability to begin the transcription of messenger RNA, which will ultimately make proteins. So, lipids as steroid hormones can communicate. Now, testosterone within men have tremendous amount of different things, functions that it does, but it can travel throughout our bloodstream to target cells and target organs throughout our body. They can communicate on a very large scale. And when it communicates, what is it actually doing? It's telling the DNA to turn on and do genetics, which is to make new proteins. Okay, that brings us to proteins. And hormones can also be proteins or protein derivatives. So, unlike lipid hormones, which turn on DNA, protein hormones do not go inside the cell. So they are going to bind to protein receptors. So the receptor is a protein, and this insulin hormone would bind to the receptor. And as it binds to the receptor, it triggers the opening of a channel which would allow glucose to get inside the cell. So glucose is too big. It can't just fit through the cell membrane. It has to go through a channel in order to get in. So it can't get in until insulin lands on a receptor and opens it up. So it's the same thing here. Insulin is the protein. Here are the receptors. Insulin does not go into the cell, but instead it lands on the receptor so that glucose can get in. Insulin is a very popular hormone because of its tie to diabetes. Again, a non-steroid hormone, which means that it's not a lipid, would hook onto the receptor and then trigger, if it doesn't open a membrane or open a channel, it can trigger a series of enzymatic reactions to actually take care of and initiate some form of communication or some form of breakdown or chemical reaction inside the cell. Okay, so talking about chemical reactions, that brings us to enzymes. Enzymes are proteins. And again, a protein is a whole lot of amino acids put together. So when we see this huge thing right here, all of these little dots would represent an individual amino acid. And the average size for a protein is 400 amino acids. And these help chemical reactions take place. And chemical reactions are the forming or breaking of chemical bonds. A lot of times we think of enzymes as digestive enzymes that break down complex food that we eat into its most small form in order to diffuse through the walls of our small intestine. But even within cells, there are many enzymes that carry out all that help carry out all the chemical reactions that take place within our body. One of the experiments we do here is we take liver, which is packed full of catalase enzyme within all the liver cells, and we mix it with hydrogen peroxide. Hydrogen peroxide is a dangerous chemical that our body naturally produces. It's a free radical or a reactive oxygen species, which would damage cell membranes. So we have all of these enzymes to help turn hydrogen peroxide into something that's safer. Does this look very safe? Well, it's just oxygen gas, oxygen bubbles, and what's left behind is water. So the chemical formula for hydrogen peroxide is H2O2, two hydrogens and two oxygens. And this is what we call our substrate. That substrate would land on the enzyme at the active site, which would help loosen up the bonds so that a chemical reaction and it can be split into different component parts. So the substrate lands on the enzyme. And again, here's another model of that. This would be like H2O2, and this would be the catalase enzyme. Obviously not to scale. But we take one thing, we help loosen up the bonds so that a chemical reaction can occur, and we can have two things left over, like water and oxygen. All right, so another form of protein are actin and myosin muscle filaments. Now, actin and myosin protein filaments exist in many types of cells, and they do play a role in cell division when we split animal cell in half and we break apart or we do cytokinesis and split it. But we most often think of actin and myosin within muscle cells. So they contract. They help pull and shorten the overall length of muscle fibers, which are muscle cells. So these protein filaments are able to pull on each other and shorten the overall length of the cell. So if all of these things contract and they all slide past each other, the length from here to here would get shorter. And when you do that with all of these individual muscle fibers and do it all at the same time, the whole muscle gets shorter. If the whole muscle gets shorter, it's going to pull on these tendons, which pull on bones and can move your skeleton. Okay, another form of protein are channels or pumps. We already talked about channels. We were talking about insulin, these extra gates to allow larger molecules across membranes. But we can also pump things using ATP. And the sodium potassium pump is probably the most common pump in your body. And it's powered through ATP. So this energy stored in this bond as we drop off a phosphate, there's energy that allows these things to get pushed across to a higher concentration. Things don't want to be more crowded. So what we need to do is force them across with the help of energy, and that's where the pumping action comes in. So, nerve impulses. When voltage is changed, channels can open. So these are voltage gated. There's voltage gated channels which allow sodium to flood into the neuron, or this axon that connects this dendrite to the sending arm of the neuron. So as we're doing this, when we send an electrical message, we're really sending that message through charged molecules or charged ions. So when we open a channel, the sodium floods in and it becomes positive, more positive here. And usually the inside of this membrane is very negative. So when you throw a lot of these positives in, you're creating a charge that can flow, which is current. But we need to reset the electrical resting state of the cell membrane. So there's these sodium potassium pumps that exist behind all along here that pushes the sodiums back out and resets the membrane to negative and resets that resting potential. So again, as far as the nervous system and electrically sending messages throughout your body, the sodium potassium pump is absolutely essential to that. So, another form of protein here are antibodies. Antibodies help fight disease, and they don't actually do the killing of the bacteria, but what they can do is they can recognize and cling to the bacteria. So we have these specialized proteins that are floating throughout our system, and if a pathogen or a bacteria that could cause disease is recognized, what happens is that the antibody would grab onto it, which would help signal something like a white blood cell to engulf and destroy that bacteria. So the antibodies go around and cling to and bind together pathogens so that the other things like macrophages can help destroy them and keep us healthy. Our final type of protein, which we don't focus on with our whole HEMPA thing, is connective tissue fiber. And the reason I bring this up, even though we talk about it more with anatomy, is that this collagen protein is the most common protein within the human body. So bones and tendons and ligaments and other connective tissue like cartilage, even looser connective tissue has these fibers in it to give it strength. So the strength of bone, the strength of tendons, the strength of um, ligaments all comes from collagen. And these collagen is kind of like rebar and cement. It adds strength to it. Big, tough fibers. Okay, let's move on to nucleic acids. So, nucleic acids. A lot of times when you hear nucleic acids, it doesn't ring a bell until you say DNA, deoxyribose nucleic acid. So, deoxyribonucleic acid is DNA, and it's made up of millions of nucleotides. And a nucleotide contains a phosphate and a sugar, which makes up this outer rung of this ladder, and then the various steps of the ladder, or the what we consider the letters of the code, are the nitrogenous bases. And in DNA there's only 4 different types of nitrogenous bases that exist: the A's, the T's, the G's, and the C's. And based off of these letters of code, it helps us actually make proteins, which we'll get to in a second here. So within our nucleus, we have the overall blueprint for every single protein that our body might need to make. So a gene is a specific sequence of letters that code for a protein. And within the human DNA, it's estimated that there's 20 some thousand different genes, but then once those proteins are made, they can be modified. So there's over 100,000 different types of proteins that can exist within any single cell. But we wind it up, because it's so long, we wind it around these histone proteins, and then we wind it tighter. And when we're doing protein synthesis and actually turning on genes, it's a little bit looser. But when we actually divide a cell and make a copy of it, they get coiled up really tightly into these sausage-like structures called chromosomes. Okay, another type of nucleic acids besides DNA is RNA. And RNA actually carries out the role of making the protein. So the DNA is the overall cookbook. It's the whole blueprint that exists within the nucleus. But then we can unzip this and take out a code for a single recipe or a single gene. And the RNA is only one strand. So again, along this edge here are the sugars and the phosphates, and then the nitrogenous bases. And again, there's 4 letters of RNA code as well, except instead of being A, T, G, and C, it's A, U, G, and C with RNA. But then this RNA now has the ability to leave the nucleus and go and find a ribosome. And at the ribosome, we read those letters of code on the messenger RNA. And then there's code because on these little guys, which are transfer RNA. And again, this is all made out of nucleotides as well. So you have this sugar phosphate backbone all along here, and there are nitrogenous bases all along here, but then these three nitrogenous bases match up with these three nitrogenous bases, and that it brings over a single amino acid, which get all connected to make up that protein. If you change the order of these letters, you change which amino acids get brought together and linked in a different order. And therefore, if you put these things in a different order, you get a whole different shape of your protein, which can then carry out a different function. So that is RNA. Again, it's a nucleic acid made out of nucleotides. We do have nucleotides that exist in ATP, and I'm going to hold off a little bit on ATP. It has 3 phosphate groups instead of the single phosphate group, and it exists as a single nucleotide. And this is a really, 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 really important molecule that we'll talk about when we talk about cellular respiration and photosynthesis, because this is how we actually pay for work to be done within living things. Okay, so those are our examples, specific examples of organic macromolecules. So hopefully that helps us give a little bit better understanding about what each macromolecule does or the different functions. All right, so that's that.